Hello, my name is Elizabeth Crowe, and my talk is about our ongoing project on ecological site concepts development for Southwest Montana using legacy data. The history of national interagency collaboration on the development of ecological site descriptions began in 2005 with an MOU signed by the USDA Forest Service and NRCS and the USDI BLM. The purpose was to coordinate development of ESDs across land management and ownership boundaries. Subsequently, an interagency manual was written in 2010 which outlines interagency workgroups and minimum requirements and content for ESDs. This was followed by a handbook written in 2013 which serves as a technical guide for ESD development. It incorporates guidance and concepts from the U.S. Forest Service Terrestrial Ecological Unit Inventory Technical Guide, as well as NRCS methods for ecological site descriptions, and clarifies the use of a two-part naming convention, one part biotic and one part abiotic. The diagram on the left shows the process for ecological site development. This diagram is from the Interagency Ecological Site Description Handbook. In the first phase of ESD development, classifications of habitat and community types, ecological site descriptions, ecological site keys, and ecological literature are reviewed to develop draft preliminary ecological sites. These draft preliminary ecological sites are then examined through multivariate data analyses of vegetation characteristics, soil properties, and site variables. Analysis will show whether the site concepts hold up, whether the current ecological site keys are reflected in the data analysis, and whether there are gaps in the data. Here's a location map for Southwest Montana showing the locations for our data. The red dots are NRCS sampling sites and the black dots are Forest Service sampling sites. We're using guidance from the Interagency Handbook for Rangelands and the U.S. Forest Service Terrestrial Ecological Unit Technical Guide to develop our preliminary ecological site description concept papers. There are three applications of our preliminary ESD concept papers: to refine current information about sage-grouse habitat on lands in southwest Montana, to provide a template for integrating ESDs and habitat types across public-private land ownership boundaries, and to provide data-supported information for further development of ESDs. We are integrating both abiotic and biotic factors in our ecological site concepts. The abiotic factors are climate elevation, geomorphology, geology and topographic and soil factors. The biotic factors are indicator plant species and plant associations. They're compensating factors that affect the distribution of plant associations across the landscape. Similar plant associations may occur on more than one soil type, as slope, aspect, and elevation or other abiotic factors compensate for differences of temperature and available soil moisture. We're organizing abiotic and biotic factors into a hierarchical structure that provides the ability to detect cross-scale ecological relationships and compensating environmental factors. Here you see an example of our draft hierarchy of ecological sites to land units. This was developed, as I talked about above, through literature review and the use of existing ecological sites and keys, vegetation classifications, and ecological literature. The third column lists two example habitat types. Within each of these habitat types are the preliminary provisional ecological sites that may occur. Now I'll talk about the second stage of our development of provisional ESDs, namely the data analysis stage. The first part of data analysis with legacy datasets is cleaning, cleaning, cleaning. Most of this involved the soils data we acquired from Surgo and Ecodata. We had to determine whether gaps in the data were because data were not recorded for a site or not entered in the dataset, or whether a site simply didn't have a particular soil characteristic. Typos and inconsistently entered values had to be corrected. All of this work ensures consistency in analysis and provides more robust outputs and associated description concept products. We supplemented the field data sets with comprehensive sets of soil moisture data, such as the relative effective annual precipitation, moisture deficit, precipitation heat load, and elevation adjusted topography, and with the normalized difference vegetation index, or NVDI, and annual frost-free days. These comprehensive data sets were helpful because of the numerous gaps in the field data. The next step was to reconcile data between the two data sets, namely the plant taxonomic nomenclature, the species abundance, the site data, and the soil properties. Reconciliation of plant taxonomic nomenclature was a labor-intensive process. Since we've moved from relying on local floras for plant names to using the National Plants Database, legacy datasets have to be brought up to date. There are two steps to this process. All plant taxonomic entities had to be labeled the same. Old names and codes had to be updated to the currently accepted plants database names and codes. Once all of the names were up to date, we combined entities wherever there would be no loss in ecological indicator value. This makes analysis cleaner and more robust. Species abundance data were collected differently by the Forest Service and NRCS. The Forest Service abundances were our canopy cover percentages. The NRCS abundances are pounds per acre of biomass. We worked hard to figure out how to equate these two types of abundances. The only possibility seemed to be regressions of cover versus biomass, but the scientific literature did not contain enough regression equations for the indicator species in our project. So we are relativizing the abundance data. Currently, we are working with the most simple relativization, namely presence and absence. We will also try relativizing by sum of the abundances for each species in each data set and by the maximum abundance for each species in each data set. Then we'll combine the two data sets. Many of the site variables have incomplete data, some of which were easy to reconcile and some of which weren't. The most difficult site variables to reconcile that we thought would be important for analysis were landscape and landform. They were based on different hierarchies and definitions for each data set, and they ultimately were not reconcilable. The soil properties data were the easiest to reconcile because all the data had been collected using standard procedures in the field book for describing and sampling soils. Unfortunately, many of the soil variables have very incomplete data, but we're doing our best to work with what we have. We have two data analysis objectives. One is to compare indicator species distribution and abundance to site characteristics and soil properties. There are two tiers of these indicators. The broad tier, which is generally referred to as series in vegetation classifications, includes such widespread western species as Idaho fescue, blue bunch wheatgrass, and mountain big sage. The narrower tier includes species that are indicators of soil and environmental conditions in specific communities, habitat types, or ecological sites. For example, needle and thread, sticky purple geranium, and saltgrass. The other objective is to compare habitat types and ecological sites to site characteristics and soil properties. There are also two tiers. The broader environment is characterized by habitat types such as Wyoming big sagebrush, blue bunch wheatgrass, and the narrow environment is characterized by ecological sites within habitat types such as shallow, limey, droughty. The analysis methods we are using start with non-metric multidimensional scaling, or NMS, using Sorensen-Bray-Curtis distance matrices for our species abundance data, or Gower distance matrices for our environmental and soil variables. The purpose of NMS ordinations is to seek patterns along the resulting synthetic axes within plot species assemblages and indicator species groups, or within assemblages of environmental and soil variables. We have started with sagebrush species, first at the series level and then at the habitat types level. We are looking for patterns in the ordination that match or don't match the provisional ecological sites within that habitat type or published ecological sites, and we're looking for environmental variables that support the patterns. We will continue our analysis by using the groupings of species assemblages and the site and soil variables that correlate to the synthetic axes to perform indicator species analysis and fuzzy set ordination, the latter of which ordinates site species assemblages while being constrained or guided by the affinity of the assemblages to predictor or explanatory site and soil variables. Other applicable multivariate analysis methods will also be used. We have some preliminary results of our first analyses of sagebrush habitat types. The NMS ordination shown on the right contains an ordination of the four big sagebrush, blue bunch wheatgrass habitat types: three-tip sagebrush, basin big sagebrush, mountain big sagebrush, and Wyoming big sagebrush. The joint plot lines show the environmental variables that are most strongly correlated to the first axis and a little to the second. The Wyoming big sagebrush and basin big sagebrush sites, which are the dark blue and light blue symbols, are gathered on the left side of the ordination and are strongly correlated with a greater number of frost-free days and a higher soil moisture deficit. The mountain big sagebrush sites, which are pink symbols, are bunched more on the right side of the ordination and are more strongly correlated with higher elevations, higher relative effective annual precipitation, and effective soil moisture combined with greenness. The second analysis slide shows a key to four of the provisional ecological sites nested within the Wyoming big sagebrush blue bunch wheatgrass habitat type. I've highlighted the soil and environmental variables in the key that are associated with the four different ecological sites. That's the red font that you see. The ordination on the left shows all the Wyoming big sagebrush blue bunch wheatgrass habitat type plots in our combined data sets. Most of them are NRCS plots. The shapes and colors of the points in the ordination correspond to the ecological site that was assigned to the NRCS plots that we received. The site comes from their Montana key. The variables correlated with the first axis are depth to the argillic horizon towards the left side of the ordination and depth to violent effervescence and thickness of the argillic horizon toward the right side. And then the variables associated with the second axis are higher elevation, depth to lithic or paralithic contact, available soil moisture, thickness of the mollicipedon, and the compound topographic index, which indicates areas of higher moisture accumulation. We will be continuing our analysis in the coming months. Thank you.